Now, Linus Manubens is going to present neuromorphological and wiring pattern alteration effects on brain function, a mixed experimental and computational approach.
Okay, thank you. Is it okay? Yeah, okay. Thank you for introducing me, and thank you for the organizers. You should be paid for that. Well, my name is Linus Manubens Gil, and I come from the Cellular and Systems Neurobiology lab in the CRG. And in our lab, we are interested in very fundamental questions in neuroscience. And in specific, in my project, we would like to know why the brain is wired as it is. The way that neuroscience has been working for many years is looking at very specific parts of the brain, teeny parts, and looking at the structure and the connectivity in these neuronal layers. What are the types of neurons that are there, if they are excitatory, inhibitory, how many contacts are they doing, and then looking at the computation that they are doing, but in very small parts. And this information is very important, but we need another layer of information, that is how the signal that is coming from one part of the brain is going to another, and how this flow of information is leading to the final function of the brain. Some ideas have been proposed in that sense, and one of the most famous is that there is a trade-off between the wiring cost of setting the network and the efficiency in processing information. If we can imagine a network that has different regions in the brain, and that we want to connect them. If we connect them with the smallest possible amount of cable, we will have that all the nodes are connected, but it has very low efficiency in processing information. In the opposite case, if we have random connections between all the regions, we will have very high efficiency in processing information, but also very high cost. What's seen in real life is that brains are in between, and this is also seen in the dendritic structure of neurons. What we want to know in my project is what parameters of this topological wiring, or what topological parameters of this wiring are crucial for brain's function. One way to address this problem is to look at perturbations of the system, and in the lab we are using both mouse models for intellectual disability and also human samples of intractable epilepsy. In the intellectual disability mouse models, for example, we have Down syndrome, where we have alterations from the synapses to the long-range connections, and importantly, in the structure of the neurons and the number of contacts that they are doing that is smaller. And this leads to learning and memory impairments. In the intractable epilepsy case, what we have is also dysmorphic neurons, altered architecture in the cortex, and also hypomyelinization. And this leads to spontaneous activity episodes in regions that are the focal regions for epileptic seizures. In order to look at the neuromorphology of these samples, as they are big samples, what we need to do is first to label the neurons, that we do using genetic tricks, and then using our black magic, that in this case is the clarity technique, we can remove all the lipids of the sample and leave the proteins in place. And then we can see both the whole picture and the structure of single neurons and, for example, the density of their connections. By the other hand, what we are doing is looking at the activity before fixing the samples, and we use electrophysiology, both with local field potential recordings that give us information of the intrinsic behavior of groups of neurons, and also calcium imaging that gives us information about how the signal is transmitted along populations. The long-term objective of the project is to introduce all this data and computational models that we developed in the lab, and to see, tuning the parameters that account for these neuromorphological properties, what is the effect of each of the alterations in the behavior of the neuronal networks. So the goals of my project are to extract fundamental laws governing neuronal wiring and understanding its optimality, to improve the understanding of the relationship between neurons, neuromorphological architecture, and neuronal activity, and to deepen the understanding of brain function in health and disease. And with that, I would like to thank my lab and all of you for your attention.